As I mentioned, today's speaker is David Liberles. He does PhD in the Durban group of Caltech working on nucleic acid chemistry. Then, doing a postdoc in the Benner group, he shifted his focus to computational methods while bringing that wealth of chemistry experience with him. Since then, he's worked on a variety of topics in molecular evolution, including work on adaptive evolution, gene tree, species tree reconciliation, and of course, all right, take it away, David.
Okay, thank you. I hope this is recording properly. And thanks for coming. As I get started, I wanted to acknowledge some of the people who've worked with me. And so a lot of this work is the work of Johan Grantham, who's in the audience. You can see him pointed to here. And so I'm normally at the University of Wyoming. I'm currently on sabbatical at the University of Canterbury in Christchurch in New Zealand. I guess you've heard some of this banter about the current state of Christchurch, but I'll give a little bit of a plug that things are starting to get back to normal. And with this, I'll turn to science and tell you about some of the things we've been up to. And so a major focus of what we've been doing is to build an interdisciplinary bridge from biophysical chemistry to evolutionary biology for the functional analysis of comparative genomic data. And our motivations in doing so are to bring a mechanistic underpinning to the analysis that the analyses that we're doing to better understand the evolution of gene functions in genomes. And so today I'm going to tell you about two different types of models that we've been working with. So one is built upon a population genetic framework that's been extended for interspecific amino acid substitution patterns. And the second is, and so this is starting from a population genetic framework. The other side of the coin is starting from a biophysical standpoint of how proteins behave as molecules and how they interact with each other, and bringing this into an evolutionary component. And ultimately, our hope is that these two types of frameworks meet in the middle, and we can have a more complete mechanistic picture of the evolution of proteins. And so part of our motivation, just to take a big picture turn for a second, is to describe the need for new models for comparative genomics. And so a lot of the approaches that we've been looking at are using methods like DNDS, the ratio of non-synonymous to synonymous nucleotide substitution rates, or fairly simple models for amino acid substitution that really simply ask the question, did a change happen or did a change not happen? And of course, the underlying biology is a lot more complex than this. And so as we start to have more complete data sets at a genomic, at a proteomic level, and start to understand some of the ways that metabolic pathways and systems behave quantitatively, we can start to build up a new framework that enables us to ask a different type of question in more detail. And so when one thinks about building up a framework for comparative genomic analysis that's rooted in underlying theory, then there are several sources of theory that we want to consider. And so population genetics and evolution give us one framework that describes the dynamics of molecules or individuals in a population. We know something about the underlying physics of proteins and biomacromolecules in general. And we also, through the traditions of Michaelis-Menten kinetics and systems biology, and we know something about how molecules interact with each other. We know something about how enzymes act and how pathways function based upon KCAT and KM. And so all of these different layers have interfaces. And so a lot of my talk will focus on this interplay between population genetics and evolution on the one hand, and protein structure and protein biophysics on the other hand. But ultimately, we want to build up a mechanistic framework that encompasses the rule set from various sources of rules in biological systems. And so this is somewhat big picture oriented. And so the next question is, what are really the goals that are a little bit more concrete that we want to focus on? And so this is phyloseminar, and so where a lot of you are interested in problems of phylogenetics. And so these are developing models that really embrace the underlying mechanistic biology of how the biomacromolecules we're using to generate phylogenies are actually behaving. And so one goal, which hasn't really been a focus of what I've been doing, but is clearly a goal in the field, is to have better models for phylogenetic reconstruction that are more robust to the underlying processes. So at a population genetic level, we have things like incomplete lineage sorting. At a structural level, there have been some very interesting papers that have come out over the last few years on that that's sort of this assumption for gene trees that you automatically have a signal that's reflective of the underlying ancestry is actually not always true. That functional convergence and functional signals can be existent in data sets, and so your gene tree may actually be a functional clustering diagram rather than something that's reflective of ancestral history. And so that's something to keep in mind as you're building gene trees. And so models that can differentiate functional signal from ancestral signal ultimately will turn out to be important. And so that's something to keep in mind, that goodness of fit and some of the more statistical measures of model appropriateness may not be sufficient for real if we're ultimately interested in biological interpretation of our trees. And then I mentioned DNDS before, and so another area that we're interested in is being able to detect functional shifts. And so a major focus of my research program has been on examining lineage-specific diversification or lineage-specific adaptations. We have a database called the Adaptive Evolution Database that characterizes these signals in a phylogenetic context. And so a lot of this right now is based upon underlying signals from DNDS or from amino acid level rate shifts. And so, but we're really just looking at rates of change, or that there is a change, and really we know a little bit more about proteins than just rates and their evolutionary properties. And so can we combine some of these properties from biophysics and from evolution to have more robust methods for understanding the functional nature of protein evolution? And then the last aspect of what we're interested in, which has also become something I've increasingly focused on, is biologically realistic simulation. That a lot of times when we think about doing analysis with real data, with real comparative genomic data, and we're interested in protein function or gene function, a lot of the times we really don't know, and annotations that are transferred by homology aren't always correct. That analyses that come from genetic studies have a fairly high level of abstraction away from the biochemical and biophysical underpinnings, and we, as you'll see later in the talk, we've recently published a paper talking about the importance of not statements in biology, and we really don't know what the interactions that are deleterious to not have are in a systematic manner that end up resulting in important selective pressures on proteins. And then we also don't know things like population size and mutation rate particularly well. And so one can, of course, do inference with very parameter-rich models, but sometimes one can actually understand the dynamics of the processes better with more control when one does simulation and can control all of the parameters of the model. And so this has been another focus, and something that I think is underestimated in its power is what we can learn from biologically realistic simulation. And so with this overview, I'm going to jump into a couple of different studies that we've initiated from these two different standpoints. And so we'll start with from the population genetic side. And so thinking about, again, building up a framework for analyzing protein sequence evolution, what are the modelable determinants of protein sequence evolution that we can play with? And so we have the mutation rate and various parameters of the mutation model, things like the transition transversion bias, which are the underlying which give us the underlying mutational propensities together with the genetic code. We have things like population size and population structure. Genes are ultimately linked together on a chromosome, and so ultimately as you'll see as we go through this, we're going to need to make more serious consideration of things like Hill-Robertson effects. And then the genes encode proteins, and the proteins form a structure, and the structure is important because it properly orients residues that are necessary for function, for example. And so binding is a function that we'll start to look at as this talk goes forward. And so this initial model that's built upon a population genetic framework is going to be fairly crude from a protein structure standpoint, and then we're going to jump very far into the other end of the spectrum with the next class of model, with the hope that eventually we can bring these two models together and really have a more mechanistically robust type of inference. And so this project was actually inspired by reading some papers that came from the protein structure community, where there's a major emphasis on the role of protein thermodynamics as a determinant of amino acid substitution. And so as an evolutionary biologist, it's fairly clear thermodynamics is important, but it's part of a larger process. And so the question is, how can we put this in the proper context? And so can we build up a model that enables us to ask how important thermodynamics actually is and to quantitate this? And it may be that the selective constraints due to protein folding are so strong that really it is all we need to consider, but we were skeptical to that proposition. And so we we've had a long history of the neutral theory, and so there's reason to be, and the neutral theory built upon data that Zucker, Campbell, and Pauling had put together. So there was good reason to be skeptical about that. So we can go back to population genetic theory, which predicts that the strength of selection, in this case thermodynamic constraint, on a protein sequence through its structure will be guided by the effective population size, where the larger the effective population size, the more power to select and the less random observed changes are expected to be. And so the idea was to see if we could put together data sets that enabled us, as well as a model to enable us to examine this question in more detail. And so this comes from a paper from Michael Lynch and Connery, and so the point of this slide, this is actually any mu, the effective population size times the mutation rate. And so the point is to show that this varies over many orders of magnitude when one goes from bacterial lineages through domesticated plants and mammals like us. And so there's a lot of scope for variation in the strength of selection. And so when we look at completed genomes, we then sought to build lineage-specific pan matrices for sets of genome pairs that had similar percent identity profiles across ranges of species that had wildly different effective population sizes. And so we could look at two domesticated species of rice. We could look at sets of primate comparisons. We could go to rodents to two Drosophila species, and then off to bacteria to two E. coli species, and where we then had the lineage-specific pan matrices that we had generated. And then from a literature search, we could then look at the average effective population size that separated these lineages. And so these are literature values of NE, with keeping in mind that bacteria are haploid and the other lineages are diploid. And so we can then use these literature values as part of the model that we're going to put together with the pan matrices that we've generated from the genomes. And so when we start thinking about how to account for protein thermodynamics, as I mentioned, we're going to start with a fairly simple in a fairly simple way. And so we're going to use the Grantham matrix. And so the Grantham matrix is derived theoretically from differences in side chain volume, polarity, and charge. And so we can use this to compare expected changes based upon these differences in thermodynamic property between amino acid side chains with what's observed from the pan matrices that we calculate. And so we can start with the Kimura diffusion equation and to look at the probability of fixation of individual amino acids. And so we can build this, and we're going to want to build this up to handle interspecific data, where we're then looking at relative probabilities of fixation of different amino acids, and where the amino acid transitions are not only characterized by their differential probability of fixation, but also by the differential mutational opportunity as determined by things like the transition-transversion ratio and the structure of the genetic code. And so we generate a mutational propensity for each amino acid transition based on that. And so as we build up this model, we're going to explicitly make some assumptions, all of which are violated, and we'll get back to these at the end of this section of the presentation. And so we're going to assume that each amino acid position segregates independently, which is a problem. We're going to assume that we have these literature-derived effective population sizes, and we're going to assume that these are fixed constant population sizes that are taken from the average of the two species that are separating between the species. We're going to assume that the changes observed are fixed rather than segregating, and we're going to assume that the transitions in each Grantham matrix category are under similar selective pressures. And so, keeping these assumptions in mind, we put together this formula for the relative probabilities of fixation, which we could then treat for every individual position, every individual transition within the pan matrices. But we can also ask in a Bayesian context how many categories are supported. where we initially tried to fit this across all of the data together, where we have, so mu j is the transitional probability, and then we have the effective population size for the pair, and then the selective constraint for either the transition, or we can bin the transitions based upon similar Grantham matrix transitions. And so when we do this, we actually have the data pushing us in a direction that leads to increasing the number of bins based upon NE and decreasing the number of bins based upon the Grantham matrix category. And so what we have here are the relative probabilities of fixation in each bin come from the observed data from our PAM matrices that we've generated. The mu's come from a simple calculation over the genetic code. The SIJs are being parameterized, and the N's are these values that we take from the literature. And so in doing this, we end up, and so I show you two models here that were among the better supported models. And so one where we have two Grantham bins, and we treat each amino acid or each pairwise comparison separately, and then the second where we have three Grantham bins and three effective population size bins. And so as I indicated, that models with more bins of effective population size and fewer Grantham bins tend to show support, and the selection coefficient, and we actually had two fairly surprising trends, that the selective coefficient always decreased with effective population size, and it decreased with Grantham value. And so these results were unexpected. And so what we were observing with the more radical changes were actually behaving more neutrally, and and we were seeing as the population size increased, that the selective coefficient on any type of transition was actually less strong, was more neutral. And so this is surprising. And so we have several possible explanations for this, and some of these deal with the assumptions that we made in setting up this model.
David, if I may interrupt.
Sure.
I'm not familiar with the Grantham bins, so when you say two Grantham bins, you mean two categories for?
Yeah, so we're reducing all the amino acid transitions are being reduced into two categories in this case, and the transitions are scored. And so the Grantham matrix gives you a score that's based upon the difference in the sum of the squares of the difference in side chain volume, polarity, and charge between two amino acids. And so that's what so this is turned into a score. This is a classic paper from the 1970s from Science that presents this matrix and this characterization. And so it would have been nice if we had support for every single amino acid transition and a lot of this complexity, but we didn't have support for that in our parameterizations of S from this model. And so we're seeing a fairly strong and so I'll get into some of the details of what's going on. So in terms of this decreasing selective coefficient with increasing Grantham value, what we believe is going on is that the more radical changes tend to be on the surface of a protein, which are more neutral, and when you have and so when you actually can accommodate changes within the hydrophobic core and more sensitive parts of a protein, these tend to be less radical changes, and so we have this position specific bias in our data that's I think leading to some of this signal, and this is something we have to control for. And so in terms of the effect with the relationship with the effective population size, that I think the strongest effect ends up being that we have this independence at the linkage level. And so there was a paper from NAVO in 1997 that suggests that when sites are linked, you can have amplifying effects that give you much stronger select effects in small population size theories than you would expect using the Chimera diffusion equation as we had. And so more properly accounting for the role of linkage at the sequence level, I think, is going to be important. So Johan's in the audience. We actually have a paper that we're working on right now that's actually looking at the link between distributions of folds in lineages and and the effective population size of the organism, and we believe that there's some signal in this data for selection on fold type and properties of the fold in different population size lineages. And then lastly, and we actually have a bias in our data that in large population size species, we're actually going to have an increasing level of segregating variation that we're averaging together with fixed changes, and that can also account for some of the effects that we're observing. And so this is now work in progress to account for some of this and see how robust our inference is to some of the assumptions that we made. And so we're developing a model that actually looks at the at these changes in PAM matrices that explicitly considers linkage, where we have both selective sweeps and background selection that are occurring in our data, and we can then look at it additively at the effects of sites that are linked to each other as we parameterize S in the model. We can also account for how much segregating variation we expect to see in our data based upon the effective population size that we're using, and so that's something that we can add. And then we can do this control where we account for protein fold and the position within defined by the solvent accessible surface area of the amino acid position, and so that's another manner to partition the data that we're considering. And of course, you asked about what we did with the Grantham matrix value and averaging together transitions. And the reason that I think there was less support for more categories is that something like a phenylalanine to aspartate change that is entirely context dependent within a protein, and so we have none of that context in our model, and so it's fairly easy to sort of average that away when we already have averaged that away in the way that we're considering it. And so we may actually have more power to enable inference at a structure-based level when we actually explicitly consider structure. And so to do so, we need to have a more sophisticated model that's also computationally efficient, so that we can do large numbers of calculations. And so that's the next part of my talk that I'm going to turn to. And so we're going to move forward into the, and so we've actually developed this in the context of simulation of protein sequences that are structurally constrained, but it's a model that can be used in a number of different contexts. And this is very much work in progress, and so we don't have all the features that we would like properly functional, and so other people are working in this area as well, as you'll see. And so there's still a lot of work to do to get these models to make these models robust. So again, when we think about how to model things, this is a little bit of a repetition in a slightly different framework. And so proteins must fold. The fold is important because it provides a scaffold for the functional residues. And then proteins, when one thinks about what is protein function is a lot of proteins simply bind to other molecules, whether they're other proteins or small molecules or RNA or DNA. And they bind as complexes, as regulators. Proteins sometimes catalyze reactions in molecules they bind. These are enzymes. And there are a few proteins form cavities to transport ions and other small molecules. And so this is another class of proteins. And of course, proteins are complex in the way that they function. And again, we're going to start fairly simply, and we'll get to some of the features of proteins that we're interested in that we may not be capturing entirely with this type of model. And so our starting point is going to be folding, and also consider binding, where we're now bringing function into the fold as part of what we're evaluating. And so when we think about, as we build up a model, how do we know that we're actually capturing the evolutionary properties of proteins? And so proteins have a hydrophobic core, they have a hydrophilic surface, and they have rate heterogeneity across sites. And typically this is evidenced in a phylogenetic context by support for a gamma distribution of rates across sites, although a gamma distribution, as many people have pointed out, is a statistical approximation for rate heterogeneity, and doesn't necessarily mechanistically capture hydrophilic core and hydrophobic core and a hydrophilic surface that are evolving at different rates, but as a proxy for doing so. And then we have typically proteins have a DN-DS ratio of around 0.2, in protein coding genes that is, where the ratio is higher on the surface than at the binding interface or the hydrophobic core. And so we expect to see these properties in our model, and we can generate a model that has these properties. We also expect to see a model where native sequences are well described evolutionarily and in their stability by the model. And so this is currently where the models that people are working with, both our models and other people's models, are breaking down, is that they're actually not describing native sequences particularly well. But it's an unsolved problem, and so we'll get to some of the reasons why and perhaps how we can move forward with this. So when we think about how proteins are modeled phylogenetically, this is the BLOSSOM 62 log scoring matrix. So modern phylogenetic models typically have site-independent Markov processes that don't consider structure and the context of substitution in any way. And so moving towards models that actually explicitly consider the structural context of an amino acid, there are two classes of models that have been employed. So they're what are called informational potentials. And so this dates back to some of the classic work from Miyazawa and Jernigan, a paper from 1985, where one looks at PDB and generates a statistical potential based upon the proximity of amino acids to each other in solved structures versus their frequency in the database as a whole. And so this has an element of structural context in it, but it averages again across context to the extent that, for example, if you have a phenylalanine and a lysine that are interacting, if they're interacting perpendicularly to each other, you have the pi orbitals from the ring that have a partial negative charge that can interact favorably with the positive charge on the lysine. But if they're interacting at this orientation, this is going to be deleterious because you'll have this is where the partial positive charge is, and so you have coulombic repulsion between these two residues, and so you lose this type of detail, and you also, for example, in disulfide bridges, which are crucial stabilizing elements, there's an angle, and of course it's a pairwise interaction, and you lose this type of nature from the informational potentials. You don't have that specificity, and so our goal was to develop a crude physical potential that characterized these underlying interatomic or interresidue processes, and our hope that really hasn't panned out yet was that if these are properly functional, we could understand protein evolution not only at the level of sequence, but also at the level of changes in underlying forces, and looking at fold transitions to understand how one fold could evolve into another fold through changes in key interactions, and also understanding how disulfide bridges can change over evolutionary time to other stabilizing interactions and some of these types of questions. But we're not there yet to answer this type of question. And so when we start to build up these models, I should give some credit to the history of where these have come from, and so a lot of this thought has come from lattice model approximations and trying to extend some of this type of thinking into real proteins. And so this is actually a figure that comes from a 2001 paper from Pollock and Goldstein, who are giving the next two lectures, and so you'll hear some more from them about this type of strategy as well. And so they've really done some of the pioneering work in the field. And so the idea is that in a lattice you have fixed defined interactions that dictate protein folding and protein-protein interaction. And so you can use these types of interactions, for example, with an informational potential, which is the way that these have classically been used, to say, okay, you have a G interacting with an I. What is that potential? And so these are then built up into a real protein, and so you can thread a sequence through a structure, and of course the distances are different. And so in the case of a pairwise interaction potential, you still have interactions with a defined distance that then can be considered to be in contact with each other, or you have the distances and you can use this coarse-grained approximation where you're then looking at the average properties of a side chain that's treated as a bead, and you can then and so this is something that also has been derived from the protein structure community, and some of the simple models for understanding protein folding that are now adopted towards protein evolution. And so I'll get into some of the terms that one can consider, both in terms of bond angles and inter-residue interactions. And so with this, one generates a contact map. And so this generates something that looks like a lattice where you can define the interactions that are then assumed to be stable through evolution with mutation and substitution. And so with this, we can look at explicit terms that come from these models that have been generated for protein folding. And so we have in this coarse-grained approximation, we have two angles that are considered, and so and these are compared to idealized values. We can look at the propensity of individual residues within the context of a stretch to be either in a helix or in a beta strand, and then we have additional terms that relate to van der Waals interactions, the classic 612 potential called the Leonard-Jones potential. We can look at the at Coulomb's law and charge-charge interactions. We can look at a hydrophobic interaction, and so looking at the hydrophobic propensity, hydrophilic propensity of a residue coupled to how solvent exposed it is. And then we've added a disulfide bridge term that's dependent upon the angle of the cysteines pointing to each other to form a disulfide bridge. And these terms all have weights that are then optimized in a fold-specific manner. And we can do something very similar with binding, where the terms that are relevant for binding are the steric interaction, the Leonard-Jones term, for bringing together the two proteins, the charge interactions that one has when one brings these interfaces together. And then as you bring residues that are on the surface together, you're desolvating them and moving them from a hydrophilic to a hydrophobic environment. And so you have a change in solvation. And so you can look at the propensity of the amino acids at the interface for this change in solvation. So this is what goes into the binding term. And so we're doing this in a coarse-grained perspective. And so this derives from a model that really dates to some of the classic work from Michael Levitt, where we have two beads. We have a C alpha bead and then a side chain bead. And then in the context of our optimizations, when we mutate we're running our simulations in a population genetic context, and so when we mutate each side chain, we have to optimize it, optimize its position. And so this turned out to be one of the slow steps in the simulations that we were doing. And so we're using a program called Squirrel, which worked in all atom space. And so my PhD student, Johan, developed a coarse-grained method that's not considering all the atoms, just in this two bead space, is optimizing the positions of the side chains. And because you're considering a lot fewer atoms in this case, it's much faster, and so it's really sped up our simulations. And so this was published last year in JME. And we're not moving the backbone. So I showed you this contact map, and so that's assumed to be fixed through our simulations, and we'll talk about that a little bit at the end. And so as we think about some of the physical chemistry that underpins the model that we're building, what we really need to consider is the fraction of time that we're spending in a folded state versus an unfolded state. It's not clear that the unfolded state is really a biologically important state, and then other states that may be more biologically important are folding intermediates, including those that can be kinetic traps, and alternative stable states. So this is the last is something that's not always real for a particular sequence, but reflects the consideration that in our question we're only asking, otherwise we're only asking, do you prefer to be in one particular state or unfolded? And so we need to ask, would you also maybe perhaps prefer to be in a different state? And so that's something that we need to consider, and that turns into one of the real problems in this method. And then binding. The unbound state actually is an important state to consider, and then also alternative interacting partners. And so this is something that This is the famous not statement. So what is if you're an enzyme, what reaction is it deleterious for you to catalyze? If you're a signaling molecule, what other partners is it deleterious for you to bind to? And so we know that specificity is one of the hallmarks of biological regulation, and so this is important. And so the solutions to these two problems end up being different, and we'll get into some of this as we move forward. And so to make a long story short, when we think about folding, because we want to exhaustively consider what are the other possibilities, because this is what the folding process is doing, at least within the folding funnel and the subset of structures that become accessible within the folding process. We use what's called the random context model, and this comes from work from Goldstein and Wolinus. And so the idea here is the sample potential contact from the amino acids in your structure, and as a mechanism for looking at the background structures that are accessible, rather than considering explicit alternative structures. And with binding, you're not potentially binding to any possible molecule, but you're binding to what's deleterious to bind to is specific other partners, and so those need to be considered explicitly. And so currently we don't know what those molecules are, but there will be a point that high throughput proteomics will get us answers to what are the relevant biological partners that are deleterious to bind to, that impose selective pressures on binding interfaces in proteins. And so this is something that we don't have the data for yet, that I think we will, and it is the more tractable of the two approaches ultimately. Explicitly, in terms of our simulations, where we're considering specificity, we're using a truncation selection scheme where the proteins have to fold and they have to bind to their native partner, and then we introduce deleterious states of binding to the wrong partner, and so this is penalized in our scheme. And so this is explicitly how we're simulating our proteins in a population genetic context, where we have explicit genotype-phenotype mappings, and we can then resample from one generation to the next, where we have explicit genotypes and an associated fitness that corresponds to folding and binding properties of the molecule as part of our simulation. So this is very time-consuming, but it gives us a layer of realism that otherwise is not accessible. So the landscape that we have turns out to be very rugged. And so when we try to optimize these terms, this is from a Sarcomology 2 domain. Some of the terms are fairly tight. Other terms, like the Leonard-Jones potential and the salvation weight, end up having a lot of fairly equivalent solutions, and the weight terms actually dictate what the idealized amino acids at positions are. And so the ruggedness of this landscape is a problem, and something that statisticians may be interested in weighing in on. But maybe this is actually a nature of the system, and something that turns into a real problem that's very hard to solve. The biggest problem, I think, is that of decoys, and so this is if we look at the sequence landscape that's close to the native states. The native states are never within or in our Sarcomology 2 domain case, are never within a nearly neutral landscape of the optimal sequences according to our model. And so at reasonable population sizes, we have directional selection to meet the specificities of the model. And so we get pushed into this part of the space from the native case. And so we know that there are some problems with packing it of the hydrophobic core in our 2-beat model, and that we don't have the volumes quite right. But given that we have very similar problems between the informational model and the 2-beat model, that leads me to think that this is actually a problem with decoys. If you look at individual protein structures, and those that are closest in structure space according to the structural hierarchical databases, and you ask, do these overlap in this close energy space? They're a little bit closer to each other in the 2-beat space than they are in the informational space, but but we don't have overlap. And so as a whole, these molecules are not informative. And what I think is the problem, and why we need to, rather than considering explicit decoys, why we needed to move to this model of random contacts, and I think we ultimately need to move to a more general version of this, is that a lot of folding is local, and so the relevant structure for one part of a molecule is not the same as the relevant decoy structure for another part of the molecule, and so having explicit decoys doesn't actually capture this aspect of protein folding, and so as we move forward and try to improve our models, we need to think very carefully about what are the relevant structures to ask are the right decoys to give us the right energy landscape, so that we're actually properly describing native sequences. And so that's an open challenge to the field. Given this, we've been somewhat limited in what we can do with these models, but we can do some things with these models. And so one of the things that we can do is we can actually recapture elements of the relative substitution rates across sites, and even elements of the specificity of sequence evolution through a fold. And so this is, again, a Sarcomology 2 domain that binds to a tyrosine-containing peptide. And so we're doing unidirectional evolution where the target peptide is not evolving, but the protein itself is. And so at the middle, we have the emission probabilities from PFAM and the relative rates of evolution mapped onto the structure. And so the bottom is the informational model. And so I mentioned the work of Miyazawa and Jernigan. These models have actually been, or these matrices have actually been updated by Ugo Pistola. And so we're using the Pistola matrices. And so in these, and so you again, in both cases, you recapture some of the site-specific rate information that's observed in the real sequences. In the 2B model, we're actually retaining some of the specific information over short evolutionary distances that is important for both folding and binding in this particular Sarcomology 2 domain. That information is actually not in the emission probabilities from PFAM, because this is averaging across Sarcomology 2 domains that have very different sequence specificities, and so that's information that should be in the top and the bottom, but not the middle. When one looks at some of the evolutionary summary statistics that I was talking about, and over longer term evolution, the top is a panel that reflects the informational models, and so you diffuse off into sequence space with these models, and in our hands, in our version of this model, one obtains support for a rate across sites for gamma distributed rates when one simulates over a phylogenetic tree, but one doesn't ever obtain support for covariant processes. The 2B model gives us support for covariant processes, as I'll show you on upcoming slides. And in both cases, we have expected patterns of DNDS evolution where the average rates are about 0.2, and the surface is evolving faster than the core and the binding interface. So these are proteins that were evolving under purely negative selection, where there's where the binding partner is retained through the simulation. And so related to this, there's actually been some discussion in the literature between Zun Gu and Hervé Philippe and others about amino acid level rate shifts and whether they're a good predictor of a functional divergence of proteins, or whether the signal is really just dictated by protein structure. And so we were in a position to look at that. And so with these simulations where we can retain the binding function, or we can change the binding function, we can ask what is the best supported model, and when do we see support for rate shifts as evidenced by support for a covariant type of model. And so the models that one can compare, one can look at an equal rates model, one can look at a rates across sites model, and then one has varying levels of complexity of covariant models, where one shifts between on and off states as well as between rate categories. And so we used Andrew Rogers' program ProCog to do this. And in doing this, when we have either negative selective pressures to retain the binding function, or positive selective pressures to change the binding function, in both cases we saw or so in the case of positive selection, we saw a faster transition from an equal rates model to a rates across sites model than when we had a purely negative selection. The transition in this particular scenario, and we actually think that aspects of this can be attributed very specifically to the mechanism, to the way that we evolved our sequences with pleiotropy constraint for multiple binding partners, is that we actually had a delay in the onset of covariant-like behavior when we had positive selection compared to negative selection. But if we look at the individual parameters, what we actually saw is we saw that this parameter actually turned out to be, this rate shifting parameter reflective of what's called the Gaultier model, actually turned out to be more important in describing these functional shifts, and these on-off categories turned out to be less important. And so we saw reduced parameterization for this parameter, but increased support for this. And so that's shown in this lower figure. And then another important point that I wanted to emphasize, and this comes from a paper we published in Proceedings of the Royal Society, is this important role of negative pleiotropy, and that it can restrict both the rates of sequence evolution as well as the capacity for functional divergence. So and so what this figure is showing is that if you have a protein that is under selective pressure to bind to one partner versus a protein that's under selective pressure both to bind to one partner and to not bind to another partner, and then you add another, and then you look at the mutational space to enable binding to a new partner, that you significantly restrict your capacity to evolve this new binding function when you also have this selective pressure to not bind to a molecule that's deleterious to bind to. And so I think this is something that sort of flies under the radar screen when we think about selective pressures due to interactions are not statements. But I think as we really start to think about biological specificity, this is going to turn out to be important. And then getting at some of the structural details of what we're observing in the case of the not statements, this is the Proceedings of the Royal Society paper. So what you see in this slide is that you have a charge-charge interaction. So you have a favorable ligand that binds here, but the unfavorable ligand is repelled by this charge-charge interaction. But you have mutations that can enable that still retain binding to this desirable ligand, but also would enable binding to this other ligand. And so you're increasing the mutational space by not having this not statement in your simulation, or if it's real data in your biological process. And so this is sort of structurally what this last figure looks like in the context of a protein. And similarly, from 2 slides ago, when we were looking at covariant behavior, this is one of the examples of 2 residues that are showing covariant behavior. And so residue 70 and residue 93, that in this case you have an arginine and a histidine. And so it's not obvious exactly what how these are interplaying, although you do have some interesting charge-charge interactions here that can modulate, as well as size interactions that can modulate the shape of this and how this positively charged residue would interact versus this serine. And so one could study this in more detail, but this is actually probably specific to our model rather than something that's real, but it shows the type of behavior that we're structurally is underpinning what we're obtaining from our simulations. And so perhaps the circuit homology 2-domain system is not convincing in terms of a major role for structural constraint. And so this part of this discussion came from a kayaking trip I recently took with Pete Lockhart, and so looking at some of the chloroplast proteins. And so I have a slide of elongation factors from one of Eric O'Shea's papers that are also show very similar types of alignments of conservation over very long evolutionary periods. And so something that really needs a structural type of model to explain properly not only the levels of conservation, but also the types of changes that are permitted. So this is in the context of translation, you have a lot of interactions, and so a lot of the surface is involved in interactions and is not able to evolve neutrally because it's critical to the function of the proteins, and so there are proteins that are even better examples of the need for dealing with structural constraint than the Sarcomology 2 domains that we've been playing with. We like Sarcomology 2 domains because of the wealth of data on the specificities of interactions that we can ultimately get into, and looking at how they change specificity and how the real proteins change specificity over biological time. So with this, the question is, where do we go from here? And so we do have this problem that our hydrophobic, and I haven't shown you the data to support this, but our 2B molecules don't have a well-packed hydrophobic cores. And so there are models out there, and so including a recent paper, Hills et al. From PLOS Computational Biology, that describe more models that have more beads in the side chains that may be interesting to play with. The other question concerns backbone movement and how much we should be optimizing a backbone. And I think we can't right now because the model has these problems of specificity. But when we start to solve some of those, we can then introduce flexibility to our backbone and add this level of realism that is known in the protein design community to be important. And then there's this sort of major question about what to do about alternative states, alternative conformations that we really need to think carefully about and play a little bit more with this random context model of Goldstein and Wolinus. And when we do get things right, I think there's a lot of using the simulation-based strategy. There's a lot of really interesting evolutionary biology that one can consider. And moving into inference, that we had a meeting recently in North Carolina for a number of us who were thinking about problems and models of this nature, and Mark Holder gave a nice presentation suggesting that some of the summary statistics that we're talking about in terms of DNDS and hydrophobic cores and evolutionary rates can be used in the context of approximate Bayesian computing, and so that's something that can be out there for people who are interested in that type of approach. And there's some other elephants in the room that I need to mention. And so indels are something that we really don't know how to deal with. So we're just talking about substitution, but when we talk about these structurally constrained models, when you delete a segment of the sequence, you need to re-optimize the packing of the secondary structural elements, and the problem becomes even worse with insertion, that you need to not only do that, but you need to model a new sequence into that as well. And so they're using things like Rosetta. One can envision how one might do this, but this is not coarse-grained, and it doesn't fit within the sort of this population genetic framework in terms of computational accessibility. And so it's something that we need to think cleverly about. Entropy is something that we're not thinking about either in the context, or some types of entropy we're not thinking about. In the context of RNA folding, we're actually working on corrections of some of the of the delta G calculations that involve a correction for the effective local concentration of one nucleotide about another nucleotide. It's not clear how this type of conformational entropy is as important in proteins as it is in RNA. Perhaps it is, and we need to be thinking about this type of correction in that context as well. Some types of interactions are not well described. So even the physics, I mentioned this cation pi interaction, and that's not specifically parameterized in our 2B model. And so the question is, how specific do we need to be? Ultimately, we also want to move into coevolution of molecules, and so we're not doing that yet, but that adds complexity to the simulations. And we're also treating proteins as static structures. We have a paper that's under review right now where we're looking in a phylogenetic context at the evolution of normal modes in proteins in some enzyme families, and so that's something that we can start to think about in this context as well, although it's also very early days for this type of sequence evolution model to also consider dynamics. And with this, I have two last slides, and so these are the two papers that sort of talk about the majority of the work that I've presented. So the paper on the left from Journal of Molecular Biology talks about the first class of models And the paper on the right from BMC Evolutionary Biology presents these structural models. And then lastly, I should acknowledge funding sources, including NSF and NIH INBRE, who've paid for the research, the Erskine program that's supporting my visit in New Zealand, collaborators at the University of Wyoming, Johan Granen in particular, who's done a lot of the work, or especially in the second half of the talk. Shruti Rastogi, who's a former PhD student of mine, who's now moved back to India, who initiated this research in the group during her PhD period. And with that, I'm happy to take any questions.